Hello, everyone. Good morning, good afternoon, and good evening, depending on where you're joining from. Thank you again for participating in GenScript GC Summit. I'm Kathy Young from GenScript, and I'll be your moderator for the opening keynote speech. We can all agree that the success of mRNA-based vaccines against COVID opened the door to a wide range of applications for this effective therapeutic modality. And beyond vaccine development, mRNA can be leveraged in protein replacement strategies, gene editing, cell engineering, and more. Today, we are honored to have invited Dr. Jason Zhang as our opening keynote speaker. Dr. Zhang is the co-founder and CEO of Zipcode Bio, a leading biotechnology company developing next-generation nucleic acid-based medicines. Dr. Zhang holds dual PhDs in immunology from New York University and chemistry from Peking Union Medical College. He completed his postdoctoral training at Yale University and Harvard University. He has a proven track record in drug discovery and development and has previously served as the CSO of 2 biotechnology companies overseeing preclinical pipelines. Next, Dr. Jason Zhang will present on some critical bottlenecks that limit the therapeutic potential of mRNA-based medicines. And the title of his talk today is Exploring the Development and Optimization of mRNA Delivery System to Advance mRNA-based Medicines. And before we start, to our audience, if you have any questions during the presentation, please type them into the Q&A box, and we will address questions during the Q&A session at the end of the presentation. So without further ado, let's get started. Dr. Zhang, the floor is yours.
Great, Casey. Thanks a lot for the nice introduction. So here I have about 30 slides. I'll go through it, and please feel free to ask questions in the chat box. The title, as Casey said, we are going to talk about how we think about advancing mRNA-based medicines to develop next generation of mRNA medicines. So this is the outline of my presentation. We're talking about how we think about the difference between mRNA vaccine and the therapeutics. And then I will introduce Zipcode Bio. I have been co-founding. I'm also the CEO. We'll basically touch on 2 major topics. The first one is machine learning for mRNA design, mainly the UTR. This was published 2 months ago in Nature Machine Intelligence by our team. The next one would be novel delivery vehicle being developed by our co-founder. We are using it actively to advance mRNA pipelines in our company. So mRNA-based medicine, as all of you must be very familiar right now, it has a huge potential. This is from a review published recently. It has outlined the major therapeutic areas and potential for mRNA-based medicines. From the top, you can see it can be used for protein replacement. And then next to it will be vaccine. Vaccine, as we know, it is approved for COVID, save millions of people's lives. And recently, Moderna has the RSV mRNA vaccine approved. In China and all over the world, people have been working really hard to develop mRNA vaccines for many other infectious diseases. On the bottom right would be cell therapy. This not only for ex vivo cell therapy, people also try to do in vivo cell therapy, especially in vivo CAR T. As example is the Capstan and other companies in the US. They are developing in vivo CAR T and in vivo CAR macrophages. And then you turn the clock, the next one will be genome editing, and then therapeutic antibody protection. Therapeutic antibody protection will be very similar to protein replacement. The difference will be it doesn't need to think about how to compete, how to show the superiority to other antibody protein therapeutics already marketed or can be developed at the same time. I will go through a few slides to explain in more details about mRNA vaccine and mRNA therapeutics, the difference. Also give you some example from our own work. So for the mRNA vaccine, let's give you an example for RSV. RSV, as you well know, it is particularly important for protecting younger child or also the senior people. For most of us, adult between these is relatively we are relatively safe, but for younger people and adult people, they are a little bit dangerous. To develop mRNA vaccines, relatively well established. Right now, people use well established AMP, SM112 or AS0315. People can do it in the lab and they can also move into a clinic, just put the antigens expressed using mRNA. But there are lots of room to improve actually. For example, the RSV, for the antigen design, how to manipulate the protein sequence, make it more stable, more immunogenic. There's quite a lot of excitement people can generate. This is some of the work. This is some of the work we did. And we filed a patent recently. This is work in my previous company. We successfully developed some RSV mRNA vaccines. The red box is our candidate. We call it M002. You can see we can use either 2.5 microgram or 25 microgram to tenfold difference dose level. And then next to the red box, you will see two benchmarks. One is from Pfizer, the other one is from Moderna. And to the far right, the blue box, that is positive control. We always use RSV-A2 inactive virus. So that is positive control. People you always include in the publication or in patent. So we can compare the result from different publication have a good sense how solid data is. This experiment was done in cotton red. Cotton red is a gold standard for RSV vaccine efficacy demonstration. So what we did is a prime and a boost, 3 to 4 weeks apart, and then wait another 3 to 4 weeks to challenge with the real RSV. Just wait a few days, we take the sample either from the nose or from the lung to look at the viral titer. As you can see here, the red box of our candidate M002, there is no viral detected, it's below the detection level. On the right, far right, the positive control also showed the completed protection. Look at the two benchmark. One of them is much better. For both of them, our candidate is way more protective than the benchmark. Based on the 2.5 microgram concentration, the protection efficiency we can get, we can estimate our efficacy is about tenfold better than benchmark Moderna's. So this, how we get such kind of good data, mainly we screened quite a lot of construct in vivo in cotton rats to see which antigen design, protein sequence design can give us the best efficacy. Even though many people are working on this RSV vaccine, there are quite a lot of possibility to improve it further. Our studies, we can get tenfold better. I'm pretty sure to improve further, there are always a possibility to do better. Now go to this slide. How do we see the mRNA vaccines? mRNA vaccines, there are a few things we always pay attention. The first one is mRNA design. I moved here because I couldn't see the screen that big. So for the mRNA, for the vaccine mRNA design, the main purpose is to express the antigen. So we don't really need it to be that high level. The carrier is well described in the literature, also proved by Moderna and Pfizer, BioNTech, either SML102 or LS. Many people can develop even superior IMPs, have a similar or even better efficacy. For mRNA vaccines, in vivo delivery, usually we just do IM injection. Of course, people are also trying to move into intradermal injection. Maybe they'll give even higher efficacy. For the pharmacokinetics, this actually for vaccine is not that critical. We do IM injection. Usually it's difficult to see because the amount of dose is so low, you don't see much exposure systemically. Clinical target, the last one, is usually to generate enough antibody response and potentially to induce memory immune response for long-term protection. So this is for mRNA vaccine. This is a review published about a year and a half ago in Nature Biotechnology. Really well summarize the key point for mRNA vaccine. To compare vaccine and therapeutics actually are quite different. So this on the right, we'll talk about the same category, but for therapeutics. Most of them are quite different, and we do need to pay attention if you want to develop therapeutics mRNAs. That is basically the next frontier for mRNA medicines. We can go through it one by one to explain a little bit more. For mRNA therapeutics, there are a few things really important to think about. It needs to express enough proteins to have beneficial effect. So to express high level, we need to do a few things, either to increase mRNA stability or increase the translation efficiency of the mRNAs. Since most of the therapeutics, for example, antibodies, those could be exogenous protein. We always need to be careful about the immunogenicity. For the does increase the protein expression level to enhance a little bit of the endogenous protein, then the immunogenicity may not be a big issue. And the second one, is a protein delivery vehicle. This one actually is quite important. Most of the time, when people try the same delivery vehicle for mRNA vaccine, they may not work that well because the LNP we use for vaccine usually they are immunogenic. Once immunogenic, then it could induce antibody or T-cell response. Then the cells, when we transfected for therapeutics, that cells could be killed, could be removed from the body. So the carrier had to be really carefully selected with low immunogenicity. And the third box, in vivo delivery, this one actually is a quite exciting area, including our company is working to that direction. I will show some data later on. For in vivo delivery, we want to target different tissues specifically. Right now, most of the deliveries go into liver. Liver is a natural sink for most of the stuff. When we inject oligos, inject anything, usually go to liver. Now people are trying to move into other tissues, for example, lung, spleen, muscle, and all other tissue type. Those generate lots of excitement. Of course, we have to be careful when we analyze those data. Sometimes it does shows artifact. And the last two also really important for pharmacokinetics. This is especially important when we try to show mRNA therapeutics can be superior to protein therapeutics. We have to show we have at least similar or better duration of action and also PK PD profile. The last one is exciting, clinical target. This we can use protein therapeutics to do the enzyme replacement. We can introduce Cas9 into the cell to do the gene editing and other gene editing mechanism. And last but not least, we can generate antibodies to show the mRNA therapeutics can be in the same area to compete with antibodies. I will show one or two slides how we have been working on the mRNA therapeutics, especially in the antibody area. So the disease we worked on is called CDI. It's a C. diff infection. It's a gram-positive bacteria. It's quite prevalent actually. It's occurring between 4 to 15% healthy adults. However, those are not that dangerous for healthy people. It is only dangerous for most of the time for senior people. 65% of the patients get infected in the hospital setting, they can relapse. So you get one infection treated well, and then it come back, and then you treat it, and it can go down, but it will come back again. Once the patient have multiple occurrence, the mortality rate is getting higher and higher. The global CDI therapeutic market is quite big. It's about $1.1 billion projection in 2027. So what we did in the lab, this is also in my previous company. It was presented last year in the third mRNA summit. The project leader actually is well. He's with us in the new company also. So what we did here, we use mRNA to express IgX. We call it IgX because it can be IgG and it can also be IgA. For IgG and IgA, we can also, just like bispecific antibody, we can also include multiple epitopes. So on the left, what you see here is survival curve. There's a lot of work going into these slides. We have a lot of data to show our mRNA encoding IgX antibodies can express antibodies secreted out from the cells in vitro to show they have the same binding activity compared to protein therapeutics. What is really interesting is in vivo, actually, by optimizing, it can show similar or even better efficacies compared to those IgG antibodies. For IgA, actually, it shows dramatic superiority because in vitro, for protein antibodies, in IgA format, it's not easy to generate. As an mRNA, is much easier. We just get all the sequence together with careful optimization, transfer into the cells. Or here we showed in vivo, it can generate potent IgA antibodies, and it is even better than the IgG regarding the efficacy. So in the middle is to show not only survival, to show the body weight. CDI infection usually induce body weight loss through diarrhea. And on the right, the panel C is basically to show the reduction of the instance of diarrhea. So this data set, even though here only showed the final efficacy, lots of work went into it, and it shows significant benefit of using mRNA to express antibodies, especially to express those antibodies that is difficult to manufacture or produce or to do quality control by traditional antibody protein therapeutics. We do have other mRNA therapeutics program before. It all presented last year in this third mRNA summit in Boston. Actually, the next one is coming next week, also in Boston. Okay, talking about delivery vehicle, this figure we also use LNP. We were working with Agitas, one of the best LNP companies in the world. Is in Vancouver, Canada. The LNP we picked has a very, very low immunogenicity. It's not the same as what we use for the RSV vaccines. And this is traditional LNP. It on the left you can see it encapsulate mRNA and deliver into the cytoplasm, and then it can express protein and either secrete out or membrane bound or cytoplasmic. It can do all the things we want. For LNP, it is traditionally made up of 4 components. It has structured lipid, cholesterol, ionizable cationic lipid, and also have a phospholipid. So these 4 components, it does give a lot of advantage for PK. It can stay in the blood for a little bit longer. However, the ratio between the 4 components is quite important. We have been working on LNPs for quite a while. We know that it has a significant advantage, also have some challenges, especially in the optimizing of the ratio, and to bypass the protection of the IP from other people. Right now, the IP situation is quite messy in the LNP field, mainly because the ratio is difficult to bypass. We touch a little bit on how we are working around this in our company. Now I would like to introduce our company. We call it Zipcode Bio. As you can imagine, why we call Zipcode, mainly because we have this technology. It can deliver RNAs into different tissues. We saw different tissues, just like the postman using zip code to deliver letters to different address. Our company, the mission is advancing next-gen RNA medicines. The company have 3 major pillars. The first one is we do have in-depth expertise in RNA and RNA-based medicine. These include machine learning-based improvement. We did publish paper I talk about a little bit later. The second one is we are using not only traditional LNPs, we are also using innovative delivery technology. Our founder called it DNP. In the company, we call it SHARP. It's much easier to remember, I would say. And the third, for pipeline, I'm not going to talk too much about the pipeline today, but we are using our technology to address unmet medical needs. Most of the pipeline are backed by our co-founders' research. By combining all those 3 pillars, our overall goal is to achieve tissue specificity, lower cost, and 4-degree stability to advance next-gen mRNA medicines. I will use the next 3 slides to give you some idea how we use machine learning to improve mRNA design. So this is a paper published in April. It's a collaboration between our company with Stanford and also Princeton University. You can see the last author, Amandi. She is a professor at Princeton University. Next one is me, and then next one, Le actually is a professor from Stanford. So when you see the mRNA, it has a cap, it has 5' UTR, it has ORF, it has 3' UTR, and it has a poly A. All those locations can be optimized significantly. For ORF optimization, people routinely do it. We sometimes also use GeneScript's platform, the free one to optimize ORF. Usually the result is quite good. It can significantly increase the protein expression. What we did in the company is focusing on the UTRs. We did a lot of work on the 5' UTR, untranslated region, also the 3' UTR. What we published is 5' UTR. On the right, this is a figure in the abstract. So I don't know if you can see it clearly. It's quite small. So roughly what we did is we pre-train the sequence, and then we fine-tune. And then we use the model, the machine learning model we generated. We do prediction to design novel sequence, and then we do experimental validation. So to put it really simple, you can certainly read the paper. There's a more detailed description of the method. Yupeng is with our company. He's the data science leader of this project. So what do we call pre-training and then fine-tuning? To put it in a simple language, when we want to design novel UTR, we have to understand how it looks like in the cell. So whatever we design, it had to look like UTR. It had to behave like UTR. That's why we call this just like a language model. Now everyone uses ChatGPT, and we can ask the AI to write essays for us. How it do it? It learned from millions of textbooks, and then know how the language, how the sentence can be written. The same thing we did here. We got millions of hundreds of thousands of UTRs from different species, and then we try to see how it looks. It has energies, free energies. It have second structures, and it have different contexts one after another. So all those things we learn, and then we know how to write a UTR. It looks like UTR, and it should behave like UTR. And then next step will be fine-tuning. How do we fine-tune? We want to know if those UTRs, it can be really translated well. So what we did is we get almost 70K UTRs have well curated translation efficiency. This is mainly by ribosome profiling. When the mRNA has a lot of ribosome on it, likely it correlated with translation efficiency. That is what we showed on the right. In ribosome profiling, on the bottom is translation efficiency. How we calculate is a ratio between ribosome profiling versus mRNA-LRF profiling. Using the fine-tuning, we can learn what kind of UTR can be translated well. And then the next step will be to really design novel UTRs. Design those not exist in the nature. And then we do very detailed validation. We use on the bottom right, we use luciferase in vitro, in vivo. We also use human EPO in vitro, in vivo to monitor the PK using the EPO protein. The cell lines we did in vitro is either muscle cell line or the liver cell line C2C12, HepG2. We did a lot of those time point study. This was done by our scientists. It's really cost quite a lot to get all the data, go back to train the model and validate all the things we have been proposed. In the end, we successfully developed the algorithm and prediction efficiency and accuracy is really good. On the right, it only showed the log for the changes of our UTR. The top UTRs we designed, this is only for 5' UTR, is easily can get about 10-fold better translational efficiency than traditional one, or even the best one people publish in the literature. So this is published in this article. You can pull out and take a look. And then talk about our delivery vehicle. This is also another really exciting area. Commonly. So we have been using LNP quite a lot. We didn't give up. We don't give up LNP because it's well established. It has gone through clinical trial. It will be foolish to completely give up LNPs. However, it does have limitations. On the left, what do we set here? As you see, it has 4 components, different colors, different shape. The problems usually, and we call it challenges, a few things. Extremely low temperature for supply chain, shipping, and storage. Typically, it's around -60. With optimization, people can improve the condition, but using the cold chain is still the safest way to do. And the second challenge is the PEG on the surface of the LNP. Before the pandemic, we had the worry if everybody exposed to the mRNA vaccine, what will happen? It's a PEG antibody is routinely discovered in the blood when people use cosmetic or all those kind of things. Once this vaccine get so well implemented, then everybody going to have the anti-PEG antibody. And now this actually is starting to become a concern in the field. More and more people start to describe sometimes the anti-PEG antibody can decrease the efficacy of the vaccines. This is how people try to either develop PEG-less LNPs, mainly to remove the PEG, or replace PEG with something else. One example is polyserine. There are a few papers published have similar efficacy or the characteristic with LNPs. And the last one challenge for LNP will be the ratio, as I mentioned earlier. The extensive optimization of the ratio among the 4 components is quite important. Even with that, it's difficult to bypass the IP war among the different players. On the right, actually, is what our co-founders have been working on over the past 3 or 4 years. What our co-founder developed is called DNP. We call it SHARP. It is a single component. How it looks like is on the right. It is have multiple designs. It has a hydrophilic part on the is blue. It also has a green part. I know it's really small. I do have the reference. You can take a look. So with hydrophilic and hydrophobic, and then it can form this kind of particle. By really smart design. It can form particle. It can package mRNA, and it does have quite a lot of advantage. I will show you later on. It have pretty interesting specific tissue targeting. The synthesis is relatively straightforward, and it showed 4-degree stability. Talk about our co-founders, Zhu and Wardrow. They are the ones developed this new delivery technology. Zhu, as everyone knows, he got a Nobel Prize last year for his contribution in the mRNA field. He's a professor at UPenn, a member of the National Academy of Science. Wardrow, he's a professor of chemistry at UPenn. He's also a co-founder of the company. He's a member of American Academy of Arts and Science. He published hundreds, close to 1,000 papers already in the superior molecular chemistry and material science. Wardrow has been working on how to mimic cell membrane using what we talked about earlier here. On the right, how to use one molecule to form a cell membrane mimic particle. He has been studying this for almost 15 years. He knows all the design, how it can form a shape that round. The story actually Zhu and Wardrow told me is about 10 years ago. Zhu has been working on NMP for a while. Anybody working on NMP, we start to realize the challenge. So Zhu really want to develop a novel delivery technology can go beyond what NMP is working, but with all the goals incorporated. So he and Wardrow they started working on this ideal design of a particle about 4 years ago during pandemic. Wardrow told us they have nothing else to do, so they plan to tackle this novel delivery technology. And what it really showed actually is this slide. So mainly on the bottom, so simple and scalable synthesis of this single component. And this can be as simple as 4 to 5 step synthesis. How it looks exactly on the bottom left, you see the green one. Green one is a phenol group, and then have 2 RO, means either or ester. The R can be a really long chain from a lipid, and on the right is the blue one that form hydrophilic part. So it can be outside of the surface, so it can interact with the blood, or inside of the particle, where it can interact with the mRNA to encapsulate the mRNA. So it looks simple. Actually, lots of know-how in it. It can form this kind of particle. The size on the right, it showed 4-degree stability. It also showed the size. The size is very similar to LNPs. Can be designed well. Can be much smaller, around 50 nanometer, or most of them actually is easily designed around 100 nanometer. On the right is showing the 4-degree stability of the particle. Here we stored in 4-degree for up to 5 months. Didn't show any changes in the particle size. We also did the expression in cells and in vivo. It shows consistent stability. Not only the synthesis and the 4-degree stability, the tissue targeting actually is also amazing. So here, show you our founder designed quite a few particle. Some of them, this is a panel of 5 different particles. It can show really nice long specific targeting, especially the last 4 there is almost no liver distribution. And these have been published on JCS, also a 5 patent. You can look up the patent number and see all the information there. No, this is a set of particles show really specific lung delivery. We also have other designs. Everything is based on the chemistry design. It can be designed to go to lung specifically, like this slide, and also like the next slide. This can show they go to spleen really well. This is an early version of the particle, and now we have a much better one. So what we published, our co-founder published, just look at the middle one, 217. This is the best among this set of particle. It shows about 85 to 90% specificity in the spleen. So you see the top is the whole mouse. The bottom is when we take the tissue out. So we can measure and compare the ratio. For others, they are enriched, not that specific. But for the middle one, 217, it goes to spleen really, really well. And now we are actively working on much better particles. So everything is about know-how. So some of the data we presented, and this is also included in the patent. Our co-founder do have all the know-how, how to design the distance between the two hydrophilic parts, and also the angle. Those will decide how tight the particle is, how stable the particle will be, and also how big the particle will be. We can make it larger, we can make it smaller. And our founder routinely do in vitro luciferase assay to increase the expression level higher and higher. Actually, by changing the structure design, recently our co-founder also published paper in JACS by manipulating the angle of the ester group there. You can easily increase the expression level up to 10 to 100-fold higher. Okay, so this is most of the delivery part we are going to talk about. And that is the end of my talk. In the end, I would just say, even though we are not talking about pipeline today, that is what we are actively developing internally. We welcome partners to work together based on our technology, the mRNA design, the delivery vehicle, and we can work on some pipeline project together, or to push the platform, get it stronger and stronger. And this is my email contact, also our CEO, Daniel Tu's email contacts. Please feel free to reach out to us. Thanks a lot.
All right. Thank you, Dr. Zhang. Very intriguing talk. And thanks for helping us understand the challenges in the development of mRNA-based therapeutics. And also thanks for sharing the innovative approaches that Zipcode Bio is currently exploring in AI-assisted UTR design as well as mRNA delivery. We will now open the floor for questions, and we already received a couple of questions from our audience. So I'm just going to read the question out, and Dr. Zhang, please help answer. The first question is, for RSV, it seems that the 2.5 microgram and the 25 microgram does not show a dose-dependent trend. Is this normal for mRNA vaccines? Thank you.
Yes, it's normal. Usually, in that experiment, we didn't show a lot of data. Probably is also included in that pattern. We have titrated down to 1 microgram and even lower, 0.5 microgram. And then we start to see a really good dose response. In mice, sometimes we start, that is actually cotton red, because in the beginning, we don't know what dose we should use. We start with high. In mice, people use 10 microgram. Actually, that usually you see saturation effect. You usually have to use 5 or 1 or maybe even lower. Then you see really nice dose response. So basically, yes, if you use 5 microgram or higher in mice, you easily get the saturation. You have to titrate it down if the vaccine candidate is strong enough. Casey, cannot hear you.
Hi, everyone. Can you hear me?
Yeah.
Okay. All right. Sorry, guys. It seems that I ran into some connection issues. Very, very sorry about that. So we will continue with the Q&A. And also, let me see. The second question is, can artificial intelligence help the process of making mRNA vaccines to be more efficient?
Yeah, we do have a strong confidence in artificial intelligence. Internally, we call it just say machine learning because we think artificial intelligence is quite a big word. But the field is moving so fast compared to 2 years ago. We started the AI machine learning project about 2 years ago with Princeton and Stanford. And we got quite exciting data, and we have a really strong confidence machine learning or AI can be used not only for UTR design, can use for the whole antigen, protein sequence design. That is into the area of protein folding antigen design. And people also start to use machine learning to design nanoparticle. And everything are going to become more and more complicated. But with the calculation power and all the chips now developed, the machine learning AI field is going to make mRNA vaccine way more potent and more safe. And yeah, this is our take.
Thank you. We have a lot of questions coming in. The next one is, what would be the targeting mechanism of SHARP for different tissues?
Yeah, that is a study our co-founder is actively working on. We do feel this is different from the LNPs. For the LNPs, go to spleen or go to lung. Sometimes one of the ideas is change the charge. Positive go to lung, negative go to spleen. In our case, it's quite different because the charge is not playing that significant role. We are doing a few pretty interesting experiments trying to figure out what is maybe binding to the particles or binding to the head of our structure. And then from there, we are trying to elucidate which one could be the key players. Our co-founder is actually working it out. If we find exciting targeting mechanism, just like liver targeting, GANET, HPR, when people know all those things, it can open some quite exciting areas for the whole tissue targeting.
All right. Due to time limit, I don't think we are able to go through all the questions, but we will have 2 more questions, and we do have a couple more so that we can address offline from Dr. Zhang. So the next question is, hi, Dr. Zhang, it's a wonderful talk. I have a question regarding the targeting strategy. Were the nanoparticles delivered intravenously when they targeted the lungs and spleen?
Yes, right now, actually, we already go beyond the IV administration. In the paper our co-founder published, they are all based on IV because those are the first step. They do the screening and trying to see if the particle can go to any tissue. The first step, they want to see if it can really be expressed in vivo, and then they were excited to see tissue targeting. All those data published is based on IV. Now we are moving beyond IV, go to much easier, convenient administration to see if we can have even better ones.
One more question. So the majority of LNP target the macrophage residing in RES organs. What makes the DNP sharp special in this case?
RES system, I think that is quite interesting. Also, sometimes when people do the tumor targeting, also rely on that. For us, we haven't really looked at systemically yet. I mean, that is really great question, and we are going to look into it.
All right. Thank you, Dr. Zhang, again for the insightful presentation, and to all the participants for joining this session. We are sorry we will not be able to address all the questions, but we will definitely reach out to you offline. So great to learn the latest development of next generation nanoparticles designed to enhance stability and organ specificity. And we do look forward to more breakthroughs in targeted mRNA deliveries and precise protein expression to maximize therapeutic impact. All right, this concludes our first talk. So please stick around as we will soon begin our next session. Please also visit our booth in the exhibit hall between each session. Thank you very much.
Thanks.